Okay, good afternoon, everybody. I appreciate we'll have some people coming through as we start. My name's Professor Dylan Clements. I'm from the Dick Vet at the University of Edinburgh, and it's my great pleasure to host this afternoon's session on genetics and host susceptibility of cats to coronavirus. So starting us off in a slight change to our schedule is going to be Dr. Christine Tait-Burkhard from the Rosling Institute, who's going to talk to us about the emergence and spread of feline infectious peritonitis due to a highly pathogenic canine and feline recombinant coronavirus. Thank you very much, Christine.
So, FCOV23, what do we know, what do we not know, and how did we actually figure this thing out? So the first thing to talk about is that we're in the alpha coronavirus family, so we're not in the beta coronaviruses. It's not SARS-CoV-2, but it is a COVID. It is a coronavirus infectious disease. Technically not wrong, of course, for the general public, that doesn't make any difference. However, in the alpha coronavirus species, we actually have at least 4 different viruses, actually even more when you count the serotypes. This is one species. It's like SARS-CoV-2, but much bigger. We're not going to go into phylogenetics here, but basically what happens time and time again with these viruses is that they're recombining. And that's why the taxonomy group said, these guys are not stable. They're one thing, they keep changing with each other. We're going to throw them all in one species. And then the different strains are different coronaviruses. And if we look here at these coronaviruses, they have an incredibly complex evolutionary history. This is actually from one of Gary's papers. And you have the serotype 1, serotype canine coronavirus, feline coronavirus. They probably came first. Some recombinations between the two led to, and mutations, led to the serotype 2, again, dog and cats. But we moved again because coronaviruses like to do one thing. They like to switch species. And we're all worried about zoonotic species switching, but equally, these viruses have caused some of the worst epidemics and of the worst enzootics and panzootics that we know. So we have to be careful that we don't neglect the economic impact that they cause for our livestock, and of course also for companion animals, because that has economic impact through the treatment costs that we associate with treating companion animals. And what has happened is that this virus jumped into pigs, as you do. And in pigs, this virus did something else. It actually acquired a partial spike gene deletion, which basically developed a natural vaccine, a mild version of the virus that was only respiratory or primarily respiratory and didn't cause any severe enteric disease as we've seen before. But we don't want to look at the entire family today. We want to look at feline coronaviruses, and we have seen this picture before. And a kind of one of the theories, as we've already heard earlier today, is that we have the internal mutation theory. So the virus is mild and benign in the cats. It acquires mutations, whether that is through environmental pressure, quasi-species development that are more favorable than others. The bottom line is that we see a change in host cell tropism that actually manages to cause more widespread disease and causes the cytokine response that is eventually problematic for the cats. As we'll hear later today, that definitely also has some association with genetics. And actually, as SARS-CoV-2 tells us, if you are more predisposed to overreact to a foreign infection, then your outcomes from infectious disease are usually worse because your body has a very strong reaction that causes damage to yourself. So we saw the press responses, and it might not be 300,000, but it's definitely also not 10,000. When we look at the stray cats responses, they lost between 30 and 40% of their shelter cats. That is a high percentage, and that is not normal. Even in a population as dense as the cats in Cyprus, even in a population that is this inbred, to a large extent, is not normal. We've seen infections in house cats, in pedigree cats. It was really not discriminating. And when we look at some of the parts of the epidemiology, and I don't want to go into too much detail because Stella is going to talk about this tomorrow, but we have seen this wave. What does the wave tell us? Well, there's a clear geographic spread which indicates horizontal transmission. Because if it's an internal mutation, why would all of these mutations happen at the same time? Why does this virus suddenly spread? Normally we say once the feline enterocoronavirus turns into feline infectious peritonitis virus, it doesn't actually shed very well anymore. You usually see kind of small isolated household infections with FIP, but it never spreads much further than that. We have seen slightly wider spreads in Japan, but nothing of this scale. Of course, again, we have to remember, cat population density will play a role into this and facilitate it, but it's definitely not the only factor. The other thing that we see in the Cyprus outbreak is the median age and the mean age. Normally, when we talk about FIPV in cats, the severe infections happen under the age of 1. If we look at the actual impact in the UK, we talk about before treatment was available, 10% of kitten deaths before the age of 1 were the result of a feline infectious peritonitis infection. That's a lot. That's 1 in 10 cats or 1 in 10 kittens, and they don't die all that easily. The other thing that we see is that we have a high percentage of neurological form in these cats. Normally we see about 14%. In these cases in Cyprus, the PCR confirmed one, we actually have 28%. That gives us causes to alarm because it could potentially indicate a change of tropism. Because you have to get to the brain somehow, or you have to spread somehow. How do you get there? I'll hypothesize at the end maybe, but a lot of unknowns that we really don't know yet. And the last thing that causes major concern with this outbreak was the first immunohistochemistry staining that we saw in the colon of these cats. Normally, and I put a comparator up, you see the staining in the villus tips, you see very isolated infection. Here you see widespread, quite damaging infection, and it looks like a different cell type. It looks potentially even like a different infection route, because it doesn't seem to be apical. It almost seems to be basolateral. So the first thing we needed to do is identify the culprit, and that has gone a little bit awry, but you can read it between the lines, between the DNA. So the problem is with feline coronavirus, isolating or sequencing from the infectious samples is not straightforward. You don't get a lot of virus, and it's usually unsuccessful without prior enrichment. So people often use a DNA bait that pulls sequences usually from the spike, but that works as long as you know what you're looking for. But we didn't exactly know what this is, so you cannot go fishing for something that you don't know what it is. Well, you can go normally, it doesn't work with DNA. The problem is also that there's not many sequences out there. They're very divergent and they're very poorly annotated, whether they're serotype 1, serotype 2, whether they're FIP, FECV, and basically we did our best, and you'll have a laugh in a second. So our plan was indeed to use basically the Arctic pipeline, with the exception that we end up with Lilo rather than Arctic at the end. So we take samples from the animals, we extract genetic material, which includes a lot of cat RNA and viral RNA. No worries, we actually tried to direct sequence, but the actual numbers of feline coronavirus sequences that we got from these samples were very clearly too low to actually do anything with, let alone to get the coverage to even just start getting a draft genome and designing primers based on that. We made cDNA because, as Al says, it's much more robust to work on cDNA, the accuracy is better, and the technology is further developed. and we amplified the genome. After that, it goes on, not to MinION actually in our case. We usually use a bigger box, that's called a GridION, just because it's more powerful. And in the end, we stitched this together with LELO. LELO works a bit similar to ARTIC, but it doesn't require as much of a reference genome to polish the sequence, which means if you have homology-directed changes or you have large indels, the pipeline's actually still able to make you a complete sequence. And the first primers we designed, we literally took FIPV sequences, aligned them, tried to get the automated primer programs to give us a primer set, which pretty much failed because it ended up with one sequence. And then it was literally hours of me and partially Amanda staring at alignments and making new primers for positions where we had a high divergence. But it wasn't successful, or it was partially successful. But as you see, the first coverage of the FCOV23 genome that we got was gappy, to say the least. Especially here, where you have spike. Let me see. That was pretty much not covered at all. And we had some coverage within the other ones. But luckily, as you can see, we have flanking sequences. And we have done some maverick approaches before. We actually have a nanopore sequencing protocol that works with 15,000 base pairs, 15,000 base pair amplicons. So something like the 3,000 base pairs, 4,000 base pairs that we needed for spike didn't really phase us. And it actually worked. We could actually successfully amplify spikes from 45 samples. We could also fill the other gaps in. And eventually, we got to a spike sequence. But when we first looked at that spike sequence and blasted it, it didn't fill us with good hope because it was canine. And it wasn't just any canine virus, it was actually a canine virus that had been associated with pantropism. Whether that's true pantropism or just kind of a good spreading virus is a good question, but it goes into many of the lymphoid tissues and the internal organs of pups and is very severe in these pups. And it first appeared in Italy in 2005, where they had a lot of puppy deaths. Further outbreaks were reported in Greece, France, Belgium, back in Italy. And it was detected all over Europe in surveillance sampling in the early 2010s. This virus has been around for a long time. So in the dog population, it's valid to even think that there's actually pre-existing immunity against this virus because it has been around Greece and potentially Cyprus for the past 20 years. So the dog population might be okay, but in the cats, we have a recombination, and that could be potentially problematic. It probably just happened at the right time in the right place with a high cat density and the right cat to jump into, and we have the problem. And when we look at the sequence, indeed, the ORF1b is canine. The accessory genes in the structural side of the things are feline, but the spike is canine. And so we also started looking into the UK cats because the UK imports several flights of Cypriot cats every week. And unsurprisingly, there have been FIP cases. The first suspect case was a very problematic one because the cat had moved through 3 different multi-cat households before it actually ended in yet another multi-cat household. Luckily, this cat only had FCOV1, traditional feline infectious peritonitis. So not too big a problem. Second cat was luckily a lucky case. First of all, the kitten in this case could be treated, but also she had a very responsible owner who kept her separate from the other cat of the household and kept her indoors. So the risk of spread in that household was very small. There was the only problem that she was in a multi-cat flight from Cyprus. However, the charity who imported the cat was not willing to cooperate with us in any way, shape or form. And we can talk about politics. I'll leave that for now or for the drink session. So when we look at the actual genome analysis, we do the classical bootstrap analysis where we can identify recombinations, and I'm sorry that this is a bit small, but we have been speaking about FCOV2, so kind of the serotype 2 feline coronaviruses, which are a canine recombinant, but the recombination is very big. It spans far more than just spike, so it has some kind of canine properties, even in the accessory genes. But what we have with FCOV23 is that it's basically completely feline, so completely adapted to a feline host in all the accessory genes, but it's acquired a new key to enter host cells. It's acquired a new spike, which could be a very golden recombination that is actually a bit reminiscent of SARS-CoV-2 and MERS as well. So this kind of recombination could be beneficial to actually enter new hosts. But if you have paid attention, you also see this little gap here. You see that some of these sequences have a deletion, and we'll go into that, and Gary will follow up with more information about that. So we looked at the spike, and the thing is, this is a serotype 2 spike. Whilst we know a lot about the mutations that are associated with FIPV in serotype 1, because that is the predominant serotype, we know very little about serotype 2. There's some mutations that are potentially associated with a change to FIP in serotype 2 coronaviruses, but none of them raised any alarm bells. There's no S1, S2 furin cleavage site in the serotype 2 coronaviruses, and there's also not in this virus. There's no furin cleavage site at the S2 prime site. It's a KRK, it's a very classical type 2 feline coronavirus, S2 prime cleavage site. So there's nothing obvious in the actual sequence that would tell us that this is readily FIP. But we can continue. We can actually look at the receptor binding domain. So for serotype 2 coronaviruses, the indicated receptor is feline aminopeptidase N. And if we model this onto the canine coronavirus that was found in humans a few years ago, then we can see some mutations that have changed compared to that coronavirus, but nothing in these upper loops that are actually strongly responsible for the binding into the aminopeptidase N. So the suggestion is that it is aminopeptidase N that is mediating fusion, and we now have data from our collaborator, David Wiesler, that confirms that this binds to feline aminopeptidase N, it binds to canine aminopeptidase N, it does not bind to human aminopeptidase N. And when he produces VSV pseudoparticles, then we actually see aminopeptidase N as a functional entry receptor. So nothing new, but actually aminopeptidase N is very widely expressed in the body. It's also highly expressed on all the monocytes, eosinophils, basophils, and NK cells, I think. So there's quite an interesting range of immune cells that express this receptor, so they express the lock that the virus can bind into, which could explain a very rapid infection and rapid dissemination. But onto the deletion. So this deletion is in the domain 0, and this is actually nothing new. We have seen this in the change from transmissible gastroenteritis virus to porcine respiratory coronavirus. Porcine respiratory coronavirus has a primary respiratory tropism with a little bit of infection in the gastrointestinal tract, but very little. In contrast to that, transmissible gastroenteritis virus goes primarily into the gut, but has a very slim respiratory phenotype. It binds sialysides and neurominidases, so it has a sugar binding ability, and that is usually associated with a change in tropism. It's also been seen in porcine epidemic diarrhea virus in the field, But there again, because porcine epidemic diarrhea virus is enteric, it hasn't really changed anything in terms of virulence. Actually, it seems to have attenuated it, but it's able to infect cells in vitro and in vivo. But it hasn't given it an advantage. So it's quite interesting how this seemingly enterically entering virus suddenly actually loses that glycoprotein. And when we look at the structure, the domain 0 sits here. So they're the little dangly bits at the end. These dangly bits actually make the spike a little bit more rigid because we also don't have an S1, S2 cleavage. And if you look here, very difficult to see, you see this receptor binding domain kind of buried behind domains A and B. This is the canine coronavirus, which has the domain 0 and the sugar binding site. If you lose that sugar binding site, it is very likely that your receptor binding site actually gets more accessible because you gain a little bit of flexibility and you gain a little bit more access to this receptor binding site. On the other hand, losing sugars, gaining sugar binding is an interesting balance that also influenza A virus, or all the influenza viruses actually strike. Influenza viruses bring their own neuraminases to actually cut themselves off from the cell surface. For coronaviruses, they have to find that balance of being retained by the cell versus gaining access, right? It's a good hook to bind to glycoproteins because it's your first hook onto the cell, and then you can tunnel to your receptor and get an entry into the cells. But once you exit, you also want to leave that cell again, so you have to cut yourself off at some point. So it could be interesting for spread. However, it doesn't seem to be that simple. If I have looked at all of the parameters, how we see long and short forms in Cyprus, there's no geographic trend. It doesn't seem to be in one area first, in another area. It's mixed throughout. When we look at the clinical presentation, it is pretty much 6.5 dozen. One of them doesn't change whether you have neurological symptoms versus the other. They both cause wet, neurological, dry, and unknown, where we have samples where we don't know. Over time, you see the percentage of long change a bit. Sorry, the percentage of short in yellow change a little bit. So there's no trend. It doesn't seem, it's associated with the epidemiology, where we have kind of the peak in the middle, where we have mostly short, but the long doesn't disappear. So there's no evolutionary advantage for one versus the other. We've also not seen both of them in the same cats. So it's not as if it were a change in the cats. We have to start comparing different sample types from the same cats. At the moment, we usually have one type. But again, it's not associated with sample type. It's not fecal. It's all through the effusion fluid, CSF fluid. It's interesting, especially once you hear Gary's talk about what this thing actually does, because it's a bit scary. So where shall I leave you with our hypothesis? So we see a fast spreading, probably more pathogenic or quicker to develop pathology, FIP version in Cyprus. It doesn't seem to be a discriminant of age, which means not discriminant of pre-existing immunity. And it's a recombinant between that feline coronavirus and the canine coronavirus. The rest of the sequences are highly identical, which is quite supportive of horizontal transmission, which we have not seen, at least not in this scale, in cats before. And we have an increase in the neurological cases. I think this will be quite interesting to see, because I think also on the physiology of cats, we possibly don't know. We know from mice, since very, very recently, that they have a lymphoid connection to the brain. For cats, nobody's looked, because we don't just do anatomy or basic anatomy like that, but actually it would be very interesting to know. So, lots of hypotheses, but lots of work to do. So what are we doing? We are currently investigating more suspect UK cases, but we also have seen an potential increase of cases in Athens, so there's more to investigate, and we have 2 pretty confirmed cases from Bulgaria. So we're going northeast of Cyprus, reasonably direct flights, dog trade going on. So it's not out of this world that this is actually spreading. We have RT-qPCR diagnostics that span kind of the second half of the spike, where you have to have a combination between canine and feline spike that works reasonably well, and we want to publish that soon. Lots of spike structure, receptor determination, virus isolation, and Volker's not here yet, but he will help us with the infectious clone construction, and all the epidemiology, pathology, following this up, because you will see what's happening at the moment. It's not as bad as it was, but it's still there. So lots of people to thank. We heard about the cats. So these are Binks and Winnie. These are Amanda's, my fabulous bioinformatician and discovery fellow. His cats. These are my 2, Artemis and Hermes. I have to mention their names because it was fate for some reason. Lots of thanks to all of our collaborators and my team. And I shall leave you with this picture for questions.
Brilliant. Thanks ever so much, Christine, for a great presentation. Can I invite questions from the audience?
Thank you, that's great, very clear. I wondered if you'd had a chance to look at any minor variants in those samples. I mean, are you sequencing deep enough to see minor variants, or?
We are in the spike for most samples, but we've not looked that deep into that yet because we wanted to make sure we have the robust full genome sequencing before we go into the depth. But yes, that is on our list to see whether there's anything popping up. The interesting bit, though, with the deletion is that that doesn't even come up in the PCR amplifications, and you think for some that would be very... well, we have over 130 spike PCRs across that deletion, and none of them have both. And you'd think in one of them it should come up if it's mixed.
That's why it was interesting. That's why it was interesting, just whether or not those variants were coming up, because I suppose you're always looking at things that are post-selection, as it were, if you're not careful, just looking at the major variants. So I was wondering if there was anything interesting going on in the background. And do you have DNDS data across those genes? Do you know if there is any evolution happening or any selection happening on those genes?
In terms of compared to other viruses or compared to...
Just between the samples. But I suppose if you don't have the minor variants, you might not have that insight quite yet.
We've not got that yet, but it's part of what we're looking at.
Eventually we got it right. My question relates to the previous question. With a lot of viruses, defective interfering particles play a major role in one of the variants showing its ugly face. Do you have any data at all that this virus actually does that? And I suppose I mentioned before the cloning sequencing. The beauty of that was that you could see quasi-species nature quite easily by sequencing different clones. You don't have that luxury here.
Yeah, no, we don't have. So let's take the second question first. So with the quasi-species, that's actually why we are doing the 15,000 base pair amplification. That's actually a 15,000 base pair virus where we're looking for quasi-species. That will get easier for this virus once it's culturable, even though you have obviously got that trade-off of culture adaptation, but it makes it easier to amplify virus and basically purify it through that. And that also then allows you to see whether you're starting to produce particles that are defective interfering. It is a proxy, but it's very difficult to assess that in an animal. You'd have to have functional partial deletion, which is a little bit trickier for coronaviruses, but there's a hypothesis that they can package subgenomic RNA and then obviously not be functional as a consequence. It's a bit like the mispackaging in influenza viruses, but it doesn't seem to be the same extent in coronaviruses.
The problem with the tissue cultures is that you don't have the selection pressure of the immune system there.
No, but equally it's an advantage because you can actually then have minor variants that actually pop their heads above water, so to speak. So it's always a trade-off.
Yeah, pros and cons, yes.
Thanks. Is this working? Is it going? Yeah. Oh, there we go. Thanks, Christine. Were any of the sequences that you have here out of cats that were non-clinical, so just FE, CV specimens, and do you have paired, like a fusion and feces out of any cats with FIP?
We do have some. They're still in Cyprus at the moment. What is interesting that actually our oldest cat, so to speak, is a December 2022 cat with FCOV23, and that cat is pretty asymptomatic. It never developed FIP, but it already has all the characteristics of FCOV23. So it's clearly not developing in all cats, but I wouldn't be surprised if all the cats in Cyprus are seropositive. But then again, kind of makes sense. If you have a 30-40% severe illness rate, that kind of seems right for a pathogenic coronavirus. Not good news for this pandemic, but hopefully good news that we can potentially develop immunity to severe infection. But again, kittens will come through again, I think, so we'll start the cycle again and again and again.
Thanks.
Thanks, Christine.